That is the end of Part B. Now look at Part C. Part C. In this part of the test, you'll hear 2 different extracts. In each extract, you'll hear health professionals talking about aspects of their work. For questions 31 to 42, choose the answer A, B, or C, which fits best according to what you hear. Complete your answers as you listen. Now look at Extract 1.
Can you tell us about the talk you will be giving at Society for Laboratory Automation and Screening 2023 and what readers can expect to learn?
Absolutely. My talk is titled A Suite of Arrayed CRISPR Assays in Primary Respiratory Cells for the Identification and Validation of New Targets for COPD. I will be discussing how AstraZeneca has invested in developing arrayed CRISPR capabilities and showcasing a project that demonstrates the potential of this technology in identifying new targets for COPD. One of the unique challenges we face is balancing the competing demands of an unbiased approach and the need for scale and automation. In the case of a whole genome screen, for instance, we use 64x384 well plates per treatment arm, which requires significant automation to make it feasible. However, when working with primary cells, it can be challenging to balance these demands while also maintaining a robust screening platform. During my talk, I will be discussing the workflow that sits behind the data, highlighting the amount of work required to set it up. I will also be sharing the additional insights that multiparametric data can provide, demonstrating the return on investment for the effort put into setting up the workflow. Overall, readers can expect to learn about the potential of arrayed CRISPR assays in identifying new targets for COPD and the challenges and solutions involved in setting up a robust screening platform for this purpose.
Can you tell us more about your research on COPD and how it could potentially lead to new treatment options for patients?
Absolutely. COPD is a chronic inflammatory disease and is currently the 3rd leading cause of death worldwide. The damage caused to the lungs during COPD is irreversible, and current treatments primarily focus on slowing down the progression of the disease. However, for some patients, the condition continues to progress, impacting their quality of life or even leading to life-threatening complications. Our research focuses on identifying new targets for COPD treatments. Specifically, we are exploring the cellular process of senescence and how it is linked to the pathology of COPD. By identifying new regulators of senescence, we may be able to develop new therapeutics to target this process, potentially offering better treatment options for COPD patients. If successful, our research could lead to improved treatments compared to existing therapies or even be used in combination with standard of care medications for better patient outcomes. Ultimately, our goal is to improve the lives of patients with COPD and contribute to better overall health outcomes.
Can you explain what further steps are necessary before this treatment can become a reality?
Absolutely. Before this treatment can become a reality, we must continue to follow AstraZeneca's 5R framework, which guides the development of any new medicine. This includes ensuring that we have the right target, tissue, safety, patient, and commercialization strategy in place. In terms of selecting the right target, we need to ensure that we have a strong link between the target and the disease. We do this by running our target identification screens in disease-relevant models, such as with our work in COPD. We then build a target validation package around strong hits and work with our therapy area colleagues to validate findings in multiple donors or even patient material. We also need to assess the target's safety profile and the commercial environment surrounding it. If all of these steps are successful, the target can enter the respiratory and immunology portfolio.
If this treatment is successful, what would it mean for both healthcare settings and patients?
If this treatment is successful, it could have a significant impact on both healthcare settings and patients. Patients with COPD would have access to a new and potentially effective treatment option, which could improve their quality of life and reduce their symptoms. Healthcare providers would also have another tool in their arsenal to treat this disease, which could lead to better patient outcomes. Additionally, if this treatment is successful, it could pave the way for further research into this area, leading to more effective treatments for COPD and other respiratory diseases.
CRISPR has gained a lot of attention in recent years for its versatility in scientific sectors. Can you explain how CRISPR can benefit different medical sectors, and are there any particular sectors you feel it could benefit?
Certainly. CRISPR is a tool that has been described as game-changing for a good reason. It has revolutionized the drug discovery process, and its compatibility with other emerging technologies, such as artificial intelligence, omics, and next-generation sequencing, has expanded its application portfolio. One of the unique aspects of CRISPR is that it spans the entire drug discovery paradigm, from identifying and validating targets to deploying CRISPR-based cell and gene therapies in the therapeutic space. In addition, we are currently exploring the use of CRISPR to identify new targets for the non-coding genome. While CRISPR screens have been widely adopted to analyze the function of coding genes, applying the technology to non-coding elements is technically more challenging. However, progress is being made, and we expect to see exciting discoveries as we start to explore the remaining 98.5% of the genome that is non-coding. CRISPR has the potential to benefit multiple medical sectors, and we are excited to continue exploring its potential in drug discovery and other medical areas.
Collaboration is often seen as critical to achieving scientific breakthroughs and technological advancements in the life sciences. How important is collaboration to your research at AstraZeneca?
Collaboration is fundamental to our research at AstraZeneca. We believe in working together, whether it's within my team, with our therapy area colleagues from different parts of the world, or through external collaborations with academic and industry partners. Our projects are part of larger matrix teams that bring together people from various parts of the business to share their knowledge and ideas. Collaboration is particularly important in functional genomics because our work requires a combination of deep disease knowledge from therapy areas and assay development and screening knowledge from our team. We are currently engaged in several external collaborations, and they allow us to explore novel science beyond our portfolio and assess new technologies. Additionally, we work closely with the Open Innovation Program, a collaboration portal that facilitates interactions and partnerships with other organizations. In summary, collaboration is a crucial aspect of our research at AstraZeneca, and we believe that it will continue to play an essential role in driving scientific advancements in the life sciences.
Scientific conferences, like the Society for Laboratory Automation and Screening, have been touted as a valuable means of fostering new partnerships and challenging new research ideas. In your experience, how important are these conferences to achieving those goals?
Conferences are an essential part of the scientific community, as they provide opportunities for scientists to expand their knowledge and connect with others in their field. The COVID pandemic has reminded us of the importance of face-to-face events, as virtual conferences can only bridge the gap to a certain extent. In my opinion, attending conferences is a fantastic way to immerse oneself in new science, reflect on one's work, and return energized and full of ideas. The connections one makes at conferences are also incredibly valuable. For instance, at a recent local meeting, I had a serendipitous interaction that helped me find a solution to a problem that had previously stumped my team. The connections and relationships one develops at these events are invaluable and can lead to new partnerships, new research ideas, and other benefits. In summary, conferences like Society for Laboratory Automation and Screening are essential for fostering scientific collaborations, challenging new research ideas, and staying up to date with the latest advances in the field.
So, what are your plans for the future of your research at AstraZeneca?
Well, I am really excited about the work we are doing in the immunology space. We have invested a lot of time and resources into developing our capabilities for functional genomic screens in this area over the past year. However, working with immune cells presents unique challenges that we need to address, such as rare cell types and donor variability. In addition, the endpoints for these screens are complex, with multi-cell type functional assays and multiparametric endpoints. Despite these challenges, I believe the potential applications of this work go beyond target identification and validation. I am also interested in extending our efforts to identify new targets for the non-coding genome. This is technically more challenging, but we have made progress, and I am excited to explore the potential discoveries in the remaining 98.5% of the genome that is non-coding. Overall, I am committed to continuing to collaborate with my colleagues, both within my team and across the company, as well as with external partners to drive innovation and scientific breakthroughs.
Now look at Extract 2. Extract 2, questions 37 to 42. You now have 90 seconds to read questions 37 to 42.
Life science research has grown significantly in the 21st century. Every year, several significant discoveries are made. Each month, thousands of original research papers are published by scientists. Although there are many causes for this growth trajectory, this article focuses on one frequently overlooked factor. The inexpensive piston pipette that can be found on many lab workbenches has greatly aided in streamlining research workflows. This article will follow the development of the piston pipette, and highlighted how air displacement pipettes from CAP can assist users in enhancing their research outcomes. Life science labs have not always used piston pipettes as frequently as they do now. For a large portion of modern scientific history, scientists pipetted by mouth. In the middle of the 20th century, a German scientist from Marburg changed this. Heinrich Schnittger created the first piston pipette by altering a syringe. Manufacturers like Kapp have worked to increase the accuracy and precision of piston pipettes since they first introduced this straightforward air displacement pipette. Modern piston pipettes have fueled the modern era's expansion of the life sciences. Air displacement pipettes are the most typical type of contemporary piston pipette. Pipettes used for air displacement have a piston that is housed inside the pipette assembly. An air cushion is used to separate the plunger from the liquid that is to be transferred. This cushion will shield samples from contamination if the user has a good pipetting technique. In early piston pipettes, the air cushion would be affected by changes in the environment, i.e., humidity, temperature, etc. Modern air displacement pipettes from Kapp can be used with high precision and accuracy in a variety of settings. Air displacement pipettes are also inexpensive and widely accessible. Positive displacement technology is used by some piston pipettes to transfer liquids. Positive displacement pipettes are excellent for transferring viscous liquids, despite the fact that they are not frequently used for the majority of lab work. The disposable tips of positive displacement piston pipettes contain the piston. In this instance, the piston and the liquid being transferred are in direct contact. Positive displacement pipettes are a fantastic option for working with corrosive chemicals and viscous liquids. However, air displacement pipettes are preferable for the majority of liquids handled in contemporary laboratories. Given the wide variety of options available on the market today, choosing the best piston pipette can be difficult. At Kapp, the hard work has already been done so that users can focus on their research while Kapp innovates the tools. All Kapp piston pipettes are created to adhere to the strict quality standards of ISO 8655 regulations. Several air displacement pipettes and a positive displacement piston pipette for challenging liquids are also part of the Kapp portfolio. The pipettes from Kapp make it easier for users to extract their data more quickly. When using piston pipettes frequently, researchers are more likely to sustain repetitive strain injuries at work. CAPP Bravo air displacement pipettes reduce the possibility of RSI with their lightweight construction and ergonomic features. The Featherlight plunger helps you apply less force to the piston pipette. Additionally, a metal shaft and double O-rings are present on the CAPP Bravo air displacement pipette's smaller volume models. With the aid of these features, CAPP Bravo air displacement pipettes can maintain their accuracy for longer periods of time and are resistant to unintentional and deliberate wear and tear. The most eco-friendly manual air displacement pipettes are those from Kapp's EcoPipette line. This green air displacement pipette is made with a longer lifespan in mind. Piston pipettes are disposed of less frequently as a result of EcoPipette's increased lifespan. This air displacement pipette is also packaged in biodegradable materials. Exchangeable volume counter mechanisms are another feature of EcoPipette air displacement pipettes. This piston pipette's cutting-edge counter mechanisms offer unmatched convenience and adaptability. The CAPP Solo range of air displacement pipettes was designed for reliable everyday use. This range's single-channel and multi-channel piston pipettes are all dependable and reasonably priced. Air displacement pipettes made by CAPP Solo are designed to maintain their accuracy and precision for longer. Users no longer need to worry about calibrating the air displacement pipettes every other week. Air displacement pipettes made by CAPP Solo have exceptional chemical and bump resistance. These piston pipettes are perfect for environments with multiple tenants because they have color-coded plungers. CAPP Trio is the only air displacement pipette in the world with many interchangeable fixed volumes. With this cutting-edge piston pipette, users will have access to 3 air displacement pipettes in one device. Simply remove the detachable volume knob and insert a different one to adjust the volume. The CAPP Trio piston pipette provides more flexibility with this straightforward volume-changing mechanism. This unique air displacement pipette offers custom knobs in addition to regular volume knobs. For additional flexibility, a variable volume pipette can be added to the CAPP Trio piston pipette. CAP microbiology pipettes defy convention by handling viscous liquids, even though the majority of air displacement pipettes are not designed to. These strong piston pipettes have no trouble handling liquids like yogurt, cream, and cosmetics. The 3 fixed volume knobs on CAP microbiology air displacement pipettes are created for the most common pipetting volumes. The CAP microbiology pipette is a straw pipette, in contrast to the majority of other air displacement pipettes. This enables users to easily and comfortably pipette food or cosmetics. If the word groundbreaking ever defined piston pipettes, it would best apply to CAPP Aero air displacement pipettes. CAPP Aero multi-channel pipettes go where no other pipette brands dare to explore. The CAPP Aero range of multi-channel piston pipettes has industry-leading channel options that help users achieve more, faster. Several pipetting errors can be traced to poor pipetting methods. When users work on their pipette handling skills, pipetting errors decrease considerably. One rapid method of setting yourself up for a good pipette is choosing an ergonomic pipette, such as the CAP Echo pipette. Utilizing an ergonomic pipette helps decrease distress and primes users for successful pipetting. CAP suggests a stable laboratory setting to decrease the chances of random error. Quality needs to be kept in mind when choosing pipette tips. Relying on high-quality pipette tips will help users to decrease pipetting errors and enhance their pipetting technique more quickly. It is also beneficial to learn to identify the various kinds of pipettes and select the best pipette for the job. One of the most efficient methods of removing pipetting errors is finding a suitable method. By learning good pipetting techniques, researchers and laboratory technicians can learn to identify sources of error in pipetting, resulting in better results for their diagnostic assays and research work. As in the case of any new skill, it is strongly recommended to practice with a reliable pipette. Users will then be able to map theory to practice and decrease pipette error while enhancing both the precision and accuracy of their measurements. There are numerous pipetting errors that several scientists are not yet aware of, which can be corrected with the correct pipette method. The first of such pipetting errors is pipetting too soon, as this may lead to systematic error. Employing calm and steady pressure on the plunger helps sustain the flow of liquid via the tip, decreasing the chances of pipetting error. Holding the pipette at an angle is another additional pipetting error common in laboratories that decreases the effective hydrostatic pressure applied to the sample volume. The perfect solution for users to tackle pipetting errors is by handling their pipette in a vertical position. One more common pipetting error is failing to pre-wet pipette tips. Pre-wetting pipette tips enables the air cushion to balance to the liquid volume being aspirated. For this pipetting error to be avoided, inserting the tip not too deeply into the liquid has been suggested, and holding it there for approximately 3 seconds before aspirating. To achieve even better outcomes, it is recommended to use premium quality pipette tips developed to decrease pipetting errors. 2 types of pipette errors cited in quality standards can impact piston pipettes. Recognizing the variation between both kinds of pipette error might help users keep their measurements truly precise. Generally, the first pipette error is referred to as a systematic error. Systematic error cites a constant variation between the volume chosen by the user, nominal volume, and the volume they dispense. The second pipette error, random error, is known as a statistical measure that outlines the possible fluctuation in pipette values due to chance events that might not be replicable every time. These include fluctuations in the pipetting environment, minor variations in the pipette, or inconsistencies in how the user has read the volume. To identify the random error, the average of 10 repeat measurements of the liquid has been evaluated, and the scatter of these measurements is identified. The bigger the systematic error or random error, the less precise a user's pipette will be.
That is the end of Part C. You now have 2 minutes to check your answers. That is the end of the listening test.